Hi everyone. We'll just wait for a few more people to join the webinar. It looks like it's settling down. Just a note, we are all recording this webinar, so it will be available later online. Anyone who does not wish to be recorded, please do note that we are recording the full meeting. So thank you for coming along to the webinar. For those of you that are unfamiliar with us, PD Frontline is a genetic testing research study, and we look at the most common genetic risk factor for Parkinson's disease, which is called the GBA1 gene. We're looking to understand this gene better and connect people from our study with other clinical trials and studies, particularly those that have a genetic focus. And today we will be hearing from Millie, who will be providing an overview of the PD Biome study. This is one of the studies that we run and work with here in London. Professor Tony Schapira will then be talking about a key publication that's come from this study, which discusses the relationship between the gut microbiome and Parkinson's disease. And then we will have a short Q&A session that will be talking about the gut microbiome. There is a Q&A button at the bottom of your screen. If you have any questions that come up for you throughout the webinar, if you could please put these in the Q&A box, and then we will get to those in the relevant sections. After that, we will move on to a brief overview of our ambassador scheme and some of the outcomes and how this is looking to change our communication with our participants. Following that, we'll have a general QA and you can ask any further questions that may have come up for you. Perfect. So, Millie, if you'd like to start.
Hi everyone, so my name is Millie and I'm a research assistant in the Department of Clinical and Movement Neurosciences at UCL. I'm one of the coordinators of PD Frontline and I also coordinate the PD Biome sub-study. So PD Biome is a clinical research study at the Royal Free Hospital, and our aim is to look at biomarkers in the gut and saliva microbiome, which might be different between people with Parkinson's disease and healthy controls. So the study involves an in-person clinical assessment and we evaluate your motor and cognitive skills. We also ask you a bit about your lifestyle, your diet, and your symptoms of Parkinson's disease. Throughout the assessment, we also go through your medical history and any medications you take. So it's a very comprehensive assessment. We ask if we can take blood, sebum, and urine samples in the assessment. This is so that we can use these samples across wide-ranging research projects. So we look from enzyme activity in your immune cells to the chemical composition of the oils in your skin. We also provide you with stool and saliva sampling kits for you to complete at home. We use these to look at both bacteria in your mouth and gut and also the metabolic products in your body. The criteria for participation in this sub-study is quite broad. So you would likely already be a participant in PD Frontline or the Rhapsody study. The Rhapsody study is for people with GBA mutations or for relatives of people with Parkinson's disease. So we ask that you are aged between 40 and 85 years old and that you're able to travel to London for the assessment. The assessments are usually around 2 hours long, so it's usually just a commitment of a morning. But if you have to travel quite far to London, we can provide accommodation for the evening before. We recruit participants from 4 health groups, so people with Parkinson's disease who have a GBA mutation, and you would know that from participation in PD Frontline or Rhapsody, where we do the genetic sequencing for this. We recruit people with Parkinson's disease of no known cause. So that's if you've been given genetic results which say that you are wild type or have no mutation. And then we also recruit people with GBA mutations who don't have Parkinson's disease. So these might be the relatives of people who have Parkinson's disease and a mutation. We finally also recruit healthy controls. So that usually is people's spouses or family members that also want to take part who don't have Parkinson's disease and have known neurological conditions. Next slide, please. So this study was first started around 2021, and since then we've gathered some really interesting data, and that's resulted in many research papers and scientific developments. When you participate in this study, you are directly contributing to ongoing research to look at the potential causes of Parkinson's disease. Some of these papers are shown on the screen, but there are also many more. These are just a select few recent ones that we've produced. So recently, we published a paper in Nature about the differences in the gut microbiome in people with GBA1 mutations. So that's the mutation that we sequence for as part of PD Frontline and Rhapsody. This has been really exciting and has garnered lots of attention for its findings, which Professor Schapira will explain next. It's just the paper that's highlighted on the right-hand side of the screen if anyone wants to read up on it following this. We're just handing over to Professor Schapira now.
Thank you very much, Millie, and welcome everybody to this webinar. So I'm going to talk about a very exciting discovery made only recently and published only just a few weeks ago, but it really made a lot of headlines throughout the world and was picked up by publications, newspapers, television, etc. From the UK right round to the US, Japan, and Australia. So really a very interesting study, and it came from your participation in this PD Biome study. We started this a little while back, and as Millie has explained, we have been collecting samples from people. In particular, I'm going to talk about the stool samples that we looked at. But it's important also to note that the information that we got from the stool samples has been related to all the other things that we've been looking at with people, such as their Parkinson's, if they have Parkinson's, or some of the other motor functions that they have, and so on. And also, we will be looking at some of the other things like blood and sebum and how they relate to what we found in the stool. So just to recap on the different groups, we had people with Parkinson's disease, and that included those with and without the GBA1 mutation. Healthy people that had no history of Parkinson's or any other neurological disorder, and also people with the GBA1 gene, but were completely healthy and had no features of Parkinson's disease. So we looked at their stool sample. And what we did with our collaborators in France was to do a very detailed analysis of the bacteria in the gut. And this particular idea of looking at the bacteria in the gut of Parkinson's disease is not completely new, but the way we did it was. And it was that revealed some very new and exciting findings. So of all of the patients, and it was almost 500 people that participated in the study, we found some very interesting and clear results in the change in the bacteria of the gut from those people who were healthy controls who were GBA negative to those who were healthy but were GBA positive, and finally to those who had Parkinson's disease, whether they were GBA positive or negative. And what we saw was this gradual evolution and change in the composition of the bacteria in the gut across the groups from healthy to Parkinson's disease. Now, interestingly, in the Parkinson's disease people, there was no difference in the pattern of bacteria between those with a GBA1 mutation and those without. They're effectively the same. But what we found in the changes that we saw was that they were not related to the drugs that people took for their Parkinson's disease, because we were able to analyze the stool in people who had never taken any medication for Parkinson's disease. They were relatively recently diagnosed. But we found that the change in the bacteria correlated or was associated with the severity of the disease. So the more severe the changes in the gut, the more severe the Parkinson's disease. But of particular interest was that in those people with the GBA1 gene, but who were perfectly healthy, we found the same Parkinson's signal, not as severe, not as significant, but nevertheless substantial changes in the gut microbiome. And then we found those changes in a small proportion of the healthy GBA negative population. So these were very interesting findings. And what they told us was that there appeared to be a gradual evolution of the changes in the gut bacteria from healthy through to genetically at risk, through to the disease itself. Now, this raises some very interesting issues, one of which is, can you use the gut bacterial signal in the people before they get Parkinson's disease to identify those who are going to be particularly at risk? Also, there are ways to change the gut bacteria. So, as we found that the more severe the changes in the gut bacteria, the more severe the Parkinson's, it raises the question of, if you change the gut bacteria, can you prevent onset, delay onset, or even reduce the severity of Parkinson's disease? Now, these are questions that we intend to answer. And I know that you're thinking, how is the gut bacteria related to diet? Well, there is a significant association with what we call the quality of diet. So a good quality diet, which contains a lot of fiber, for instance, fruit and vegetables, a lot of unsaturated fish oils, and so on, as opposed to a very bland and highly processed diet. So a good quality diet is associated with less risk of Parkinson's and less severe Parkinson's disease. And I know you might wonder, how can the bacteria in the gut influence Parkinson's disease? Well, the bacteria in the gut produce a lot of chemicals, and some of these chemicals can increase the production, for instance, of inflammation in the gut and in the blood and potentially in the brain, and also can increase the level of alpha-synuclein, which you know is an important protein in the cause of Parkinson's disease. So there are very clear and logical reasons why the content of the gut microbiome might influence the onset and course of Parkinson's disease. But as I just mentioned, there are many questions which this important study now raises and which we want to try and answer. And we want to use the same PD Biome study to try and answer those questions. Things like, do the gut microbiome changes remain over time, or do they change over time? Can we influence the gut microbiome by intervening with different diets? And so on. I should add that the changes that we saw in the people that come to our London microbiome study were also seen in the colleagues that we collaborate with in Italy, and also in other studies that have been undertaken in the United States, Korea, and Turkey. So there's clearly a global influence of the gut microbiome on Parkinson's disease. And we intend to investigate this further to try and use it to better understand Parkinson's disease and to try and maybe treat it. So I'm going to stop there. And maybe if there are any questions that people would like to ask, I'm going to be happy to answer those now. And then we'll move back to our agenda.
Thank you, Professor Schapira. So if anyone has any questions that you'd like Professor Schapira to answer, please could you just add them into the Q&A box at the bottom. There's been a few asking about how to get involved in the PD Biome study. So you can email us to express your interest, and I'll send you the participant information sheet and a bit more about what your participation would entail. And we'll send that email address in the recording of this webinar following this. We have a question from a viewer for you, Professor. So given that spouses' diets are likely to be similar to the person with Parkinson's disease, is there a problem with using spouses as controls?
Yeah, that's a very good question. And the answer is no. We are comparing the healthy controls with the Parkinson patients and looking at the contrast between the two diets. And then we will look at those people, for instance, the spouse control, we'll look at their microbiome and how it relates to the microbiome of other controls without any Parkinson and without any Parkinson spouse. So the information is really still very important. And we know that, of course, people who live together generally tend to have the same diet, but that's not a problem for us. In fact, in some respects, it's an advantage.
Thank you very much. There's a few more coming in. So there's a question asking, does it help to take probiotics?
Another very good question. I don't know the answer to that yet, but that's certainly one of the questions we want to answer with our study. So the first part of continuing our study is going to look in more detail at the microbiome, and then we're going to start looking at the effect of probiotics and see if they change the Parkinson microbiome. Now, it's going to be important to change the Parkinson microbiome in the right way, not just in any way. So I can't say for sure yet whether probiotics are a good thing or a bad thing, because we'll need to know how they change the microbiome and whether they increase the good bacteria and decrease the bad bacteria, or alternatively do the opposite. And if they do the opposite, of course, that's not a good idea. So a very good question. That is certainly on our radar, but I think we probably need to do that a little bit further down the line when we know more about the microbiome in Parkinson's.
Thank you very much. Can alcohol consumption adversely affect the microbiome?
Yes, very much so. But I can't tell you for the moment whether it's going to affect the microbiome in Parkinson's disease in the right way or the wrong way. So at the moment, I can't give you specific advice on alcohol consumption, but it certainly does influence the microbiome generally. There's no specific association of alcohol and Parkinson's disease other than the general advice about not consuming too much alcohol.
Thank you. We have a question asking if there is any relationship between gut microbiome and dopamine.
Yes, there is. Another very good question. So, some of the bacteria in the gut can increase the metabolism of levodopa and dopamine, and significantly affect the potency of some of the medication. And this has been used in the past to try and improve the efficacy of some of the medication used in Parkinson's disease. Although I think it's proven to be rather difficult to sustain, and other ways to do that, like combining things with the levodopa, have proven easier. But yes, the gut microbiome can influence dopamine. And it can do so in a positive or negative way, depending on the composition of the microbiome.
Thank you very much. There's a question here saying, you mentioned gut bacteria changing across all groups in the study. How do you know these changes are specific to Parkinson's disease and not a generalized change?
Okay, so the changes that we saw didn't occur across all groups. So in the perfectly healthy controls with no GBA mutation, the change in the proportion of patients was quite small, only about 4 or 5% of the healthy controls. But remember, Parkinson risk is about 4 to 5%. In the GBA population, we saw about 20%. But those with the microbiome changes were those, interestingly, with some very subtle changes that suggested they might be at greater risk of Parkinson's disease. So the finding that we had was an evolution of the microbiome from healthy control to Parkinson. It didn't occur in the same proportion across all groups. Very uncommon in the healthy controls. In the minority of the GBA positive but no Parkinson group, and then in the great majority of the Parkinson group. So it clearly was a Parkinson signal and very specific.
We have a few questions about fermented foods, things like kimchi. Do you recommend that similar foods can be eaten daily to help with Parkinson's disease?
I think my advice from the evidence we have at present is that the more varied a diet is, the more rich it is in fiber, the better it is. And I think we can say that that probably runs across most healthcare related dietary issues. So I'm not familiar so much with kimchi, but if it's got a lot of fiber in it, then it's likely to be a good idea. I note that somebody is asking about short chain fatty acid supplementation. And this is a good question because, of course, the gut bacteria produce short chain fatty acids, and the type that they produce and the volume that they produce is very important in influencing the lining of the gut. At the moment, I can't advocate any particular supplementation with short chain fatty acids. We need to know more about what the Parkinson microbiome is producing in terms of short chain fatty acids. So can't answer that question at the moment, but it's obviously a very good one.
Perfect. We'll take a few more questions before moving on. So there's a question about if there's any relation between celiac disease and Parkinson's disease.
None that we know of at present. The microbiome may be altered in celiac disease, of course, but we don't know whether that will lead to any relationship with the Parkinson microbiome.
And there is a question asking, could antibiotics be used to regulate gut bacteria populations?
They can be, but it's not the best way, because the effect of antibiotics is relatively short-lived. So after a few weeks, the microbiome tends to revert back to what it was before the antibiotics. The best way to change the microbiome is through diet.
And perhaps a final, or one of the final questions now, does the microbiome influence the onset of Parkinson's disease, or is it Parkinson's disease that influences microbiome?
From the data we have in our study, it's the microbiome which influences the onset of Parkinson's disease, and influences the onset in terms of the increase in the risk and potentially the age of onset and rate of progression. However, as I've emphasized, we need to know more about this, which is why the PD Biome study is so important to us now, that we've got this really exciting result. And we're so grateful to the people that are signed up, but we need as many people as possible to sign up. If people have been in the PD Biome study already, your data are probably in our publication, and we're really desperate to find out what happens to the PD Biome over time. So you're particularly going to be helpful to us, but all other people who are first to come to the PD Biome study will also be really helpful. So a big plea here to help us answer these really important questions.
Perfect. And perhaps a final question. What about fecal transplants?
Yes, a very good question. So fecal microbial transplants, as they're called, are already used in human disease. A particularly bad intestinal infection with a very nasty bacteria called Clostridium difficile, that if it doesn't respond to the normal routine antibiotic therapy, fecal microbial transplant can be used from human to human. And it's very simple. It's a simple dried tablet of the bacteria that's taken. So that can change the PD microbiome potentially. And that is certainly on our radar to use in the future in a clinical trial. But we need some more basic information first before we know how the FMT trial could be designed and used. But a very good question.
Thank you very much, Professor Schapira. We'll probably end the Q&A there. Thank you very much. But just to kind of reassure everyone, if there's any outstanding questions that you've asked, we can answer these following the webinar when we send out the recording. And we'll also send the link for where you can contact us for the PD Biome study if you are interested in signing up. And from there, we'll send you further information, such as the participant information sheet. So keep an eye on your emails following this, because the full recording of the webinar will be sent then, as well as any additional answers to outstanding questions and any additional information that you might need. So we'll pass back on to Georgie, who's going to talk a little bit about kind of the ambassador scheme and communication updates from us.
Hi everyone. Thank you for contributing all those questions to that Q&A. That was very interesting for me as well. So this section is a slight diversion from the topic we've been talking about. It is about our participant engagement and outreach as well, and generally how we're going to be managing our communications going forward. So we've set up an ambassador scheme for our study, which has 8 people, mostly with Parkinson's and also supporting family members. who have joined our efforts to improve the PD Frontline experience for participants and extend the reach of our research. So we have 166,000 people in the UK with Parkinson's, and less than 5% of those have joined our research. So we're accessing a really small number of people with Parkinson's. So part of the role that our ambassadors help is to support our outreach. So they share information about our study with people in their community, and they help connect more people with Parkinson's to research. They also check our participant-facing documents, so any newsletters that we share, any documents that you would sign, like a consent form, go through our ambassadors, and they check that the information is accessible and what we're sharing is of interest. They also give us feedback on things we might not have considered, and this can be anything from the size of a button to information that would be useful for people with Parkinson's. And there have been a few key takeaways from the ambassador scheme so far. The ones that we've been focusing on the last 6 months have mostly been to do with signposting, communications, and sample updates. Next slide, please. Great. So signposting to relevant support networks and resources on our website. So our website is full of lots of different information about the work we do, some of the trials and studies that we work with. But one of the key themes that came out of our first ambassador meeting was that feeling alone and isolated with a Parkinson's diagnosis is quite common. So we've set up a page on our website to provide some information about groups that are providing support for Parkinson's. So if you want to have a look at these will have some links and contact numbers for relevant charity groups and other support services. Some of our ambassadors also kindly offer to talk to individuals with particular questions about living with Parkinson's and registering to our study. So we are looking to develop some signposting for after you find out your results as well, to put them into context about what research they are relevant to. In terms of sample updates, this highlighted for us that when you're waiting for results, the more information we can share, the better for you. So we now understand the importance of sharing why the genetic testing process can take such a long time, and we are taking the opportunity in this webinar to share that with you. So our testing process now takes about 6 to 8 months to complete on average, which is a great improvement from the 1 to 1.5 years that we were seeing this time last year. And each sample goes through several stages of testing. So there are many different points at which the testing can be waiting. So one of the biggest waiting points is the DNA sequencing step. So this is the part where we test to see whether there are any variants within the GBA1 gene. And this step is highly complex and requires some supplementary testing to confirm that all the things we are finding are accurate. The other key waiting point is after posting your sample. So we have to wait for batches of about 95 samples to arrive so that the machines that do the testing can process your samples. So this is one of our larger waiting points, and we thought it was important to share with you why your sample can take so long to get back to you. But this length of time is varied, and we're currently working on methods to provide you with brief updates in between when you send your sample to us and when you receive the result, just so that you know that we're still working on it. The other main outcome of these scheme has been about communication. So that's where this webinar comes into place. Our ambassadors have recommended increasing frequency of communications, providing more accessible information and signposting and summarizing some of the research outcomes that we're producing and any of the groups we're working with are producing. So following this feedback, we've started to increase some of our informative communications, and we now share newsletters every 3 months, which focus on updates from our study, updates from associated studies like the PD Biome study, some of our associated trials, and also spotlights on some of our participants and the groups we're working with. We intend to run 6 monthly shorter webinars covering information about our work like this, and some of the research group outcomes that we're working with too. So we have put up a poll towards the end where you can give your feedback and select which topics would be of interest to you for a future webinar. Then we can look to tailor this towards your interests. So this is just an overview of some of the work that we've been doing to improve your experience on the study and keep up to date with our work on Parkinson's. And we are going to move on to a Q&A session. So if you do have any questions that have been unanswered, we will look to go through those now. If your question doesn't get answered, we will provide a document on our website which will have answers to any questions that don't get answered in this webinar. Okay, so the slide on the screen is just some information about our social media and our website. So if you do want to get in touch with us, please use the information here to get in touch. Great. So let's have a look at the Q&A. Okay, so we have a question. Is there any correlation between B12 deficiency and a more at-risk gut biome or linked to Parkinson's disease? Dr. Toffley, do you have any thoughts on B12 and Parkinson's disease?
Hello, not to my knowledge, though I'm not an expert in gut microbiome. I don't know if Professor Schapira is still here.
He's left for other work. So
no, to my knowledge, there's no correlation between B12 deficiency and gut microbiome.
Okay. So someone here said they've sent in samples for us for the genetic testing. Does this mean that they can't take part in the microbiome study until they have the results? So you're welcome to express an interest. We can make a note on your account. In terms of being eligible for it, we would have to know whether you have a genetic change or you don't have a genetic change in order to be eligible. So for now, we would say wait until you get your genetic results. And when these do come through, we'll have a note on your profile, but equally do get back in touch with us and we can look at setting up an appointment. And we have a question from one of our ambassadors saying about, would you like us to encourage people to sign up for the PD Biome study, as well as the wider PD Frontline study? So yes, if it's something that you would be able to explain to any of your surrounding community, that'd be great. This is a really important study for us, and it's something that we do look to identify people in our PD Frontline cohort who might be eligible for it. Equally, we do have a lot of people on the study, so when we know someone's particularly interested, that does help move things along a little quicker. So yes, if you would be happy to share that we do run a sub-study based on the gut microbiome and that's of interest, do give them our details and we can have a further chat with anyone who is interested. So we have a question here. Perhaps you could answer this one, Dr. Toffley. This participant said they have a GBA gene with a variant. Would their daughter have it too and their grandchildren? How would this affect family?
Yes, thank you. So yes, if you know you carry a GBA variant, as with any other genetic variant, really, your risk of passing it to one of your childs is 50%. So without knowing anything else, your childs have a 50% chance of carrying the same variant that you do each. If any of your childs carry this variant, they in turn will have a 50% chance of passing this variant to their child. The risk of getting Parkinson's in people that carry one of these variants is variable, and most variants increase the risk only by a small amount. Most have an increased risk that goes between 3 and 5%, most of the variants in Europeans at least. So what is the exact risk in people carrying this variant depends on the specific of the variant. And if you have received the results through us, we should have provided you with the risk estimate associated with carrying this variant. If you also require more specific counseling regarding your genetic status, you can just write an email to us and we can set up a call or reply via email so that you can have a more personalized genetic counseling.
Thank you very much. On a similar note, we have a question about the LRRK2 mutation and whether this is being explored differently to the GBA mutation.
So LRRK2 is another gene that is associated with Parkinson's. And the short story is that variants in the LRRK2 genes are increase the risk of Parkinson's more than GBA variants. People, just for reference, people that carry a variant in LRRK2 have a 80% chance of getting Parkinson's in their lifetime. So it's a significantly higher risk. We only test for some specific LRRK2 variants. And so the way we test for them is different from how we test for GBA. In the general population, LRRK2 variants are a lot less common than GBA. And same in Parkinson's. If GBA variants are found in around 10% of people with Parkinson's in the UK, LRRK2 is found in around 1%, perhaps even less. So they are different genes that affect the risk in different ways. And we do test for them differently as well, although we do it through the same samples that you provide to us.
Thank you. And this question asks which LRRK2 variants we test for. So we do test for the most common LRRK2 variant, which is the G2019S variant. Okay, and then we have a question asking about the link between ADHD and PD. Is there any genetic link with ADHD and PD?
So genetic link, again, I'm not the word expert of genetics of the ADHD, but to my knowledge, nothing definitive has been described. Obviously, as who asked the question probably knows, ADHD is also associated with dopamine dysregulation, although in a different way and in different parts of the brain. And so it's reasonable to question whether Parkinson's and ADHD are linked in some way. But as much as we know, the answer is no so far. The cognitive deficit that some people with Parkinson's develop has some resemblance to ADHD in the sense that attention and working memory seem to be affected in a predominantly in people with Parkinson's, but that's as far as the relation goes, as far as I'm aware, at least.
Thank you. Millie, this one might be for you. So this person's asked about GBA1 and LRRK2 and whether having both makes you eligible or ineligible for the PD Biome study.
Thank you. You would still be eligible. We would class you as a GBA1 PD participant, but also we would make sure to note that you also have a LRRK2 variant. It's always interesting to see people with different types of variants and gather data from them to see, any kind of results that come from that. So you would still be eligible. If you would like to contact us about that, then we can provide you with a bit more information. There's also a question here, Dr. Topley, if you would like to answer, do people with young onset Parkinson's have more gut-related problems?
Again, I might not be the best person to answer this. People with young Parkinson's, so I can give an indirect answer. I don't, to my knowledge, the direct answer, so measuring the gut microbiome in people with early onset Parkinson's has not been done yet. What I can answer is that people with young onset Parkinson's, and by young onset, usually we define people that develop Parkinson's before the age of 50 or before the age of 45, according to the definition. And these people usually carry some specific genetic risk factors for Parkinson's more frequently than people that get Parkinson's later in life. For example, genes like PINK and PARK2 are more and Parkin are more often associated with young onset Parkinson's. And we know that people that carry these genetic variants have a specific microbiome signature. So that we do know, but the direct answer is not known, or at least not to my knowledge. I apologize with the audience if I don't know the specific answer, but as you can imagine, this is quite a broad and novel topic, so I might not know all the flavors of it.
That's perfect. Thank you. So I can see we're getting to the end of the webinar. So anyone who's not had their questions answered, please be assured that we will go through these and put them on our website in a Q&A document towards the start of next month. We will be putting up a poll which should appear on your screen. It's a couple of questions. The first is about whether you found the content interesting, and this second is about whether you would like certain topics to appear at future webinars. So anyone who is able to vote, we would greatly appreciate your feedback, and then we will try and tailor future webinars towards topics of interest. So I'll just give you a moment to fill those out.
In the meantime, as well, just in case anyone missed any of it earlier, if you are interested in participating in the PD Biome study, we'll send the email address when we send out this recording of the webinar, and you can contact us through that for a bit of extra information and just to express your interest.
Perfect. Well, I think we'll close the webinar there. Thank you everyone for coming, and everyone who has participated in the PD Biome study so far. We hope to see some of you take some interest and join our study going forwards. We will keep an eye on our mailbox for anyone who is looking for more information or looking to get involved. So thank you very much for attending. We will share this recording on our website.